My name is EVANS OTIENO Achola. Today I'm going to take you through blood smear, preparation and staining. You are welcome. By the end of this lesson, you as the learner, you should be able to explain the purpose of preparing blood films. The second objective, you as the learner, you should learn techniques used in preparation of thick and thin blood films, and you should also be able to prepare a standard thin and thick blood smears. Okay, we are going to start by defining some basic terms like a blood film or a peripheral blood smear is a thin layer of blood smeared on a microscope slide and then stained in such a way to allow various blood cells to be examined microscopically. The aim of blood smear. The first aim of blood smear is to investigate hematological problems (disorders of blood cells). The second aim is to look for parasites within the blood cells, such as malarial parasite and filarial parasites. Okay, we have two types of blood smears. The first one is a thin blood film and the other one is thick blood film. In thin blood film is prepared from anticoagulated blood obtained by venipuncture or fingerprint prick blood, mainly used to study various blood cells. The second blood film is the thick one, which is prepared from a drop of anticoagulated blood placed on a microscope slide and slightly spread to form a thick layer of blood on the slide, mainly used in diagnosis of blood parasites, particularly malaria. Let's look at the procedure for making the thin blood film. You place a drop of blood at the 3/4 end of the clean frosted microscope slide. You place another slide or spreader with smooth edge at the angle of 45 degrees near the drop of the blood. Then you move the spreader backwards so that it make contact with the blood. Then you rapidly move the spreader forward over the slide. A thin peripheral blood film is thus prepared. Then you dry it and stain it. The diagram below this diagram here shows the procedure for preparing the thin film. This is a pictorial showing the difference between a thick and a thin smear. This one is a thick smear, as you can see. Then this one here is a thin smear. Our second type of smear is the thick blood smear. This one you made when you place about 2 microliter of blood in the center of a clean microscope slide. Using a thin glass rod, you spread the blood in a circular area of about 1.5 centimeters. Then you dry the smear, then you stain. This one is a pictorial representation of how thick smear is made. Let us continue with the thick blood smear. After preparing it, you air dry the smear. You fix using methanol or you spray easy fix for 5 minutes. Then you air dry the smear. Let's look at the staining procedure. For thin smear, flood the slide using field stain. B for 5 seconds, wash with running water, flood with field stain. A for another 10 seconds, wash with running water again, air dry and view under times 100 microscope lens using oil emulsion. For thick smear, flood the slide using field stain A for 10 seconds, wash with running water, flood with field stain B for 5 seconds again, then again you wash with running water, you air dry, then you view under the microscope. The below pictorial demonstrates the fixation and staining procedure of thin and thick smears. From this picture you can see this is a thick smear and this one is a thin smear. Here this is a thin smear as viewed under the microscope, and this one is a thick smear as viewed under the light microscope. Today's class, thank you so much for attending and being attentive.